All right, everybody, get ready because today we're going to be doing a deep dive that's going to be like a biology class just for you.
Yeah.
So we're going to be talking about DNA replication. We're going to be looking at how we go from, one single fertilized egg to the trillions of cells that make you.
Right.
And we're going to be doing this deep dive using some excerpts from a biology textbook on DNA replication. And it should be fun.
Absolutely.
Yeah.
So if you think about it, you started off as just one single fertilized egg, like a tiny little seed
Wow.
that grows into a mighty oak tree.
Hmm.
And now every single cell in your body contains a complete copy of your DNA,
That's amazing.
your own blueprints.
And it's got to duplicate that every time like a cell divides.
Exactly.
Yeah.
Every time a cell divides, it needs to make a copy of that whole instruction manual.
Gosh, that's a lot.
So that's where DNA replication comes in.
Okay.
Make sure that when a cell divides, each new cell gets a perfect copy of the genetic instructions.
So we start with one, and we end up with two that are the same.
Exactly.
Oh.
And each new DNA molecule that's made consists of one original strand from the parent molecule
Okay.
and one brand new strand.
Oh, so it's not just like a photocopy?
It's not a straight copy, no.
It's like,
Think of it more like passing down a treasured family recipe
oh,
to the next generation.
okay, yeah,
You want to make sure it's accurate.
that makes sense.
So to understand how this works, we need to look at the structure of DNA itself.
Okay.
DNA is shaped like a twisted ladder
Okay.
or a double helix,
Ah.
like a spiral staircase.
Great.
And each rung of that ladder
Yeah.
is made up of two base pairs.
Okay.
And they're very specific.
Uh-huh.
Adenine always pairs with thymine,
Okay.
and guanine always pairs with cytosine.
It's like a little puzzle.
Yeah, like a matching game.
A with T and G with C.
Exactly.
But how does this twisted ladder get copied? Like, how does that actually happen?
So imagine a tiny zipper running along the DNA molecule. An enzyme comes along and unzips the DNA,
Oh.
breaking the bonds between those base pairs.
Wow.
And then another enzyme comes along and brings all these free nucleotides,
Okay.
which are like the building blocks of DNA.
Like the little pieces.
Yeah, they're raw materials.
Yeah.
And it attaches the free nucleotides to each unzipped strand.
Okay.
And it builds two new strands that are complementary to the original ones.
Wow.
So A always goes with T and G always goes with C.
Yeah.
And in the end, you have two identical DNA molecules, each
Yeah.
with one old strand and one new strand.
That's amazing.
Like I said, it's not a photocopy. It's more like...
Yeah. Yeah. It's more complicated.
Yeah.
That's really cool.
It's elegant.
It is elegant. Yeah.
Yeah.
And I've heard about mutations, right?
Yes.
Aren't those like errors that can happen
Yeah, mutations are errors that can occur during DNA replication.
during this process?
Yeah, during this whole process.
Do they always cause problems?
Well, that's a great question.
Yeah.
And the answer is not always.
Okay.
Some mutations can be harmful and can lead to diseases.
Right.
Others can actually be beneficial
Oh, wow.
or even have no noticeable effect at all.
So it's like a typo
Yeah,
in the DNA code.
that's a good way to put it.
And sometimes it's just a minor error and it's fine.
Right.
But sometimes it can change like the entire meaning of the sentence.
Exactly.
Yeah.
Some mutations can actually give an organism a survival advantage.
Wow. So it can actually make it better.
Yeah, it can actually make them better adapted to their environment.
That's incredible.
It's pretty amazing.
So like a typo that actually makes the poem better.
Yeah, like a happy accident.
Yeah.
But of course, there are also mutations that can be detrimental,
Right.
leading to problems in an organism's development or function.
So it's like a game of chance. Some are good, some are bad, some are neutral.
Exactly.
Wow. So understanding the different types of mutations is really important then.
It's crucial
Yeah.
for understanding how genetic information is passed down and how variations arise in populations.
This is getting really interesting. Can we dive into some specific types of mutations?
Absolutely.
I'm eager to learn more about how these typos can actually affect the code.
Let's do it. Okay, so let's start with base pair substitutions.
Okay.
Imagine one base pair in the DNA sequence being replaced by a different base pair.
Okay.
Like instead of an A pairing with a T, it gets swapped with a G and a C.
Okay.
It's like changing a single letter in a word.
Yeah, I can see how that could change the meaning.
Right.
Like, does that always have a big impact though?
Not necessarily. Sometimes a base pair substitution might not even change the amino acid sequence in a protein. Or the change might be in a non-critical region.
Okay.
You know, proteins are the workhorses of our cells.
Right,
They're
yeah.
carrying out all sorts of functions.
Yeah.
So even a small change in their amino acid sequence can have an impact,
Okay.
but it's not always the case.
So it's like a typo that, like, sometimes it changes the meaning of a sentence. But sometimes it's just a spelling error and, like, you still get the message.
Yeah, exactly. Now, frame shift mutations are a bit more dramatic.
Okay.
Imagine one or more base pairs being added
Okay.
or deleted from the DNA sequence.
Okay.
And remember how the DNA code is read in groups of 3?
Yeah, the codons, right?
Exactly, codons.
Uh-huh.
Well, adding or deleting bases shifts that whole reading frame.
Oh, wow.
It's like if you were reading a sentence and suddenly a letter was added or removed.
Oh, yeah.
The rest of the sentence wouldn't make any sense.
No, be all messed up.
Exactly.
Okay.
So frame shift mutations are much more likely to lead to a non-functional protein.
It's like scrambling it up.
Yeah, the instructions get all scrambled.
It can't be made.
And the protein can't be made properly.
So base pair substitutions is like changing a letter. And frame shift is like adding or deleting a letter.
Yeah.
So it, like, really messes up the message.
Exactly.
Are there other types of mutations?
There are other types, but these are two of the most common important ones.
Okay.
Now, these mutations can happen spontaneously during DNA replication.
Right.
But there are also external factors that can increase the likelihood of mutations.
Oh, like what?
They're called mutagens.
Mutagens.
Things like UV radiation from the sun
Oh, right. Yeah.
that can damage DNA.
Yeah. Remember learning about that.
Exactly. So when UV light hits our skin cells, it can cause something called thymine dimers to form.
Thymine dimers.
Yeah.
Okay.
So remember how we talked about base pairs?
Yeah.
Adenine always pairs with thymine in a DNA molecule.
Right.
Well, when two thymines that are next to each other on a DNA strand get hit by UV radiation, they can actually bond together, forming a thymine dimer.
So instead of, like, pairing up with the adenine on the other side, they stick to each other.
Exactly.
Okay.
And this creates a kink in the DNA molecule, like a bump in the road. And this can disrupt the DNA replication process and increase the chances of a mutation happening.
So it's like the thymine dimer throws a wrench in the whole thing.
Exactly.
And then it's more likely to make a mistake.
Yeah, and that can lead to things like skin cancer.
Which is why you got to wear sunscreen.
Exactly.
Yeah.
But let's shift gears a little bit.
Okay.
Let's talk about how DNA actually carries the instructions for building proteins.
Okay.
Because DNA itself is like a blueprint.
Right.
But it doesn't directly build anything.
Oh.
It needs a messenger and a translator.
Oh, a messenger and a translator. Okay.
Yeah.
So who are they?
Well, the messenger is a molecule called messenger RNA, or mRNA for short.
mRNA.
And the translator is another type of RNA called transfer RNA, or tRNA.
tRNA.
Oh, okay.
Okay, so we've got mRNA and tRNA.
Right, and it all starts with a process called transcription.
Transcription, okay.
During transcription, the DNA code is copied into a molecule of mRNA.
So it's like a copy of the blueprint.
Yeah, that's a good way to think about it.
Okay, why do we need a copy, though? Why not just use the original DNA?
That's a great question.
Yeah.
The DNA is precious.
Okay.
It needs to be protected.
Right.
It stays safe and sound in the nucleus of the cell, which is like a vault.
Like the library.
Yeah, exactly.
Right.
The mRNA, on the other hand, is like a mobile messenger that can travel out of the nucleus into the cytoplasm, where the protein-making factories are, which are called ribosomes.
So it takes the instructions from the DNA in the nucleus out to the ribosomes.
That's exactly right.
Okay.
So next comes translation.
Okay.
So the mRNA code is actually read and used to build a protein.
Okay.
And remember the codons.
Yeah, the 3-letter groups.
Yeah, those 3-letter sequences that code for specific amino acids. Well, the mRNA carries those same codons.
So each codon says, okay, use this amino acid.
Pretty much. So in the ribosomes, the codons are matched up with tRNA molecules. Okay. Each tRNA molecule carries a specific amino acid.
So the tRNA is like the delivery truck.
Yeah, like a little delivery truck.
Okay, that makes sense.
And it brings the amino acid to the ribosome. And then they get all linked together.
Oh, cool.
To form the protein.
Like an assembly line.
Exactly like an assembly line.
That's pretty neat.
Yeah, it's pretty amazing.
The mRNA is like the instructions. And then the tRNAs are like bringing the parts.
Yeah.
And the ribosome is like putting it all together.
That's a great way to visualize it.
Yeah.
And this whole process from transcription to translation is happening constantly in our cells.
Wow.
Producing all the proteins that our bodies need.
Okay, now this is like blowing my mind a little bit.
Okay.
Because if DNA is like the blueprint, how come like different cells in our body have such different functions?
That is a great question.
Don't they all have the same DNA?
They do.
How can a muscle cell and a brain cell be so different if they have the same, like, instructions?
That's where the concept of gene expression comes in.
Gene expression?
Yeah.
Okay.
Even though all of our cells have the same DNA, not all of the genes are expressed in every cell.
Wait, so genes can be, like, expressed or not expressed?
Exactly. Gene expression refers to the process by which the information in a gene is used to create a functional product, like a protein.
Oh.
And that process can be turned on or off in different cells depending on their function.
So it's like having a giant cookbook. But each cell only uses the recipes it needs.
I like that analogy.
Oh.
That's a great way to think about it.
Yeah.
For example, the genes that code for proteins involved in muscle contraction are expressed in muscle cells, but not in skin cells.
Right.
And the genes that code for proteins involved in producing melanin, which is the pigment that gives our skin its color.
Right.
Those are expressed in skin cells, but not in muscle cells.
Okay.
So this selective gene expression is what allows different types of cells to specialize and do their unique jobs.
So they know which genes to use.
It's remarkable, isn't it?
It is.
This control of gene expression is complex.
Yeah.
And it involves a lot of different factors.
Like what?
Well, signals from other cells, environmental cues, and even the cell's own internal state.
Okay.
All these things can affect which genes get turned on or off.
So it's not just the DNA, it's like the DNA plus everything around it.
Exactly.
That makes it what it is.
Yeah, it's like this beautiful dance of molecules and signals that shapes the diversity of cell types in our bodies.
Wow, this is so cool. So DNA replication and mutations and gene expression, it's all connected.
It's all part of this intricate web that makes life possible.
It's awesome. Wow, so we've talked about so much already, like from DNA replication and mutations and gene expression. We've really zoomed in on how these molecules are working. But I'm ready for more. Like, what else can we learn about DNA?
Well, let's go back in time and explore how scientists are using DNA technology to unlock secrets of the past.
Ooh, like the mummies.
Exactly.
Yeah, we talked about that before, analyzing the DNA from mummies.
It's amazing.
It's mind-blowing how we can actually learn about, you know, ancient people and extinct species just from their DNA.
It really is like a time machine.
Yeah.
And one of the most powerful tools in this field is ancient DNA analysis. Where scientists can extract DNA from ancient remains, like bones and teeth. And then they use techniques like PCR to amplify and analyze that genetic material.
So they're basically like piecing together a puzzle from these little fragments that survived.
Like a genetic jigsaw puzzle.
Yeah.
And by comparing ancient DNA to modern DNA, scientists can learn so much about evolution, migration patterns, relationships between different species. They've even traced the evolution of humans going back thousands of years.
So we can actually see like how human DNA has changed over time.
That's right.
That's incredible.
And it's not just limited to humans. You know, ancient DNA has also been used to study the extinction of mammoths and other Ice Age animals.
Oh, wow.
Really shedding light on what led to their disappearance.
So it's like we're unlocking this, like, hidden library of information.
Yeah.
Genetic history.
Exactly.
Like, what other secrets do you think it holds?
Well, who knows?
Like, can we bring back a mammoth?
Maybe.
Like in Jurassic Park?
Well, bringing back extinct species is still science fiction.
Yeah.
But some scientists are actually exploring that idea.
Wow.
I mean, think about it. It would raise a lot of ethical questions.
Oh, yeah, for sure.
But it just shows you the power of DNA technology.
Yeah.
And what we might be able to achieve in the future.
This has been amazing. I feel like I have learned so much in this deep dive. Like, we went from a single cell to like the entire history of life on Earth.
It's all connected.
Yeah, it really is. I feel like I've got a new appreciation for all those little processes that make life possible. So thank you.
You're very welcome.
And to all our listeners out there, I hope you enjoyed this deep dive into DNA as much as I have.
It's fascinating.
It really is. And who knows what's coming next?
Absolutely.
Until next time, keep those